Hello students, ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. So welcome to Sai Niketan online biology classes. So this is the 10th day of sexual reproduction in flowering plants. Yes. So ಇಲ್ಲಿವರೆಗೆ major heading cover ಮಾಡಿದ್ರೆ pre-fertilization structure and events ನಾವು cover ಮಾಡಿದ್ದು. Fertilization we covered in the last video. So just post-fertilization structure ಬಗ್ಗೆ ನಾವು ತಿಳ್ಕೊಂಡಿದ್ವಿ. But elaborate ಆಗಿ ಈ ಮುಂದಿನ ವಿಡಿಯೋದಲ್ಲಿ ನಾವು ಕಲಿಬಹುದು. So fertilization first we understood. So almost we covered this one. So fertilization ಏನಾಗುತ್ತೆ ಅಂತ ಹೇಳಿ ಒಂದ್ಸಲ ನೋಡ್ಕೋಬೇಕು. Fertilization otherwise a double fertilization heading ಇದೆ ಅಲ್ಲಿ. Double fertilization means it is a unique phenomenon that occur only in the angiosperms. ಈ double fertilization ಅಲ್ಲಿ ಎರಡು event ಆಗಲೇಬೇಕು. So after pollination, the pollen germination takes place. Pollen tube growth occurs through the stigmatic surface and stylar region. Ultimately, pollen tube enter into the ovule. Yes, either through micropylar end, that is a orogamy, otherwise from the chalazal end, that is a chalazogamy, or from the lateral side, that is a porogamy. So ultimately, ಈ pollen tube micropylar region ಎಲ್ಲಿ synergid ಇದೆಯಲ್ಲ ಅಲ್ಲೇ deviate ಆಗುತ್ತೆ. So it is guided only due to the chemotrophic ovule. Clear? So it come near the synergid one. As it strike the synergid, so pollen tube burst ಆಗಿ ಎರಡು ಮೇಲ್ ಗೆಮೆಟಲ್ ರಿಲೀಸ್ ಆಗುತ್ತೆ. Similarly, the synergid also ruptures. Yes. So the finger-back projections which are present here, that is a filiform apparatus, that will guide the entry of the pollen tube toward this particular region. So ಎರಡು ಮೇಲ್ ಗೆಮೆಟಲ್ ರಿಲೀಸ್ ಆಗಿದೆ. So this red color is a egg or oosphere ಅಂತ ಹೇಳ್ತಾರೆ. So this is a central cell with the two polar nucleus, green color dot in them. Blue color is a two male. Yes, both male gametes are produced from the generative cell. So ಈಗ double fertilization ಎದಕ್ಕೆ ಅಂತ ಹೇಳ್ತೀವಿ. So this is a red color is an egg. This red color is nothing but egg or sphere of the term. This blue color is a one male gamete. Yes. So this the egg plus male gamete unite. That is actual called as the syngamy or generative fertilization. That is actual fusion between male and female gamete. So this is a one event. So ಇನ್ನೊಂದು event ಆಯ್ತು. So this is the first event. So next one, another male gamete fused with the central nucleus. Means before the union of this male gamete and this one, these two green color structures. So these are the polar nucleus or the central nucleus combined from the secondary nucleus. ಎರಡು ಸೇರಿಸಿ ಒಂದು secondary nucleus ಇರುತ್ತೆ. This secondary nucleus or polar nucleus plus male gamete. Blue color is the male gamete. So ಎರಡು ಸೇರಿಸಿ, so the secondary nucleus is a diploid one due to fusion of two nucleus. Male gamete is a haploid one. So collectively three nucleus are formed. This is called as a triple fusion or vegetative fertilization. Yes. So this is the second event. ಇದು ಎರಡನೇ event. Both events take place inside this embryo cycle. That's why double fertilization is a unique phenomena seen only in the angiosperms, which include two events. One is a syngamy where the male gamete fused with the egg or the oosphere. Another male gamete, this is called the syngamy of generative fertilization. In another event where the male gamete fused with the secondary nucleus or polar nucleus from the diploid structural form, that is called the triple fusion. Either product to either product. So the product of syngamy, is called as a zygote or oospore ಅಂತ ಬರಲಿ. The product of triple fusion is a primary endosperm nucleus. So this will develop into primary endosperm nucleus. The product of C may be developed into zygote. So this is about the double fusion, means double fertilization. Now after fertilization, the next topic is a post-fertilization and structure. In last video we studied some chart. Yes, fertilization and post-fertilization. Fertilization and structure develop event. So we will take this one. So briefly, one tabular column will come over. After fertilization, the calyx, corolla, stamens, stigma and style, everything will fall off. They will wither. Yes. Now come to the internal structure. The ovary develop into fruit. Yes. The ovary wall develop into fruit wall, that is called the pericarp. Next, inside, the ovule develop into seed. Yes. The integument of the ovule turn into testa and tegment. Outer integument develop into testa, inner integument develop into tegment. Yes, sometime in seed, the nucellus may be there, that is a persistent nucellus, that is called the perisperm. Yes, so next, these are the major changes. Ultimately, the ovary turn into fruit, ovule turn into seed. So these are the main structure. Now, this zygote later on. So zygote develop into embryo. Yes. The primary endosperm nucleus, which is present inside the primary endosperm cell. So this is the central cell, yes, which contain polar nucleus. For means triple fusion takes place in that cell only. So now this primary endosperm cell, in which primary endosperm nucleus is there, this the complete structure develops into endosperm. Yes, this primary endosperm nucleus along with the primary endosperm nucleus develop into endosperm. So these are the two structures produced from this structure, means inside the embryo cell. Along with that one, so we are going to cover what are the seeds, what are the fruits and its typification. Means ultimately, in post-fertilization structure and events, we are going to study first endosperm formation. Second one is the development of embryo from the zygote or the oosperm. That is the second event. That is also called the embryogeny. Then collectively the ovule develop into seed. Types of seed we are going to cover. Then the fruit also develop. So these are the major heading: endosperm formation, embryo development, seeds and the fruit. These are the topics which are going to cover under this heading: post-fertilization, structure and event. So first one is the endosperm. That one we are going to cover. So post-fertilization structure, first one is the endosperm. Yes. So again, so this is a secondary nucleus. This is a male gamete combined to form primary endosperm nucleus. Yes. So after fertilization, whatever the event occurs, this embryo sac develop into mature embryo sac, in which here, so these antipodal cells degenerate. So whatever the three cells are there, so these are the degenerating antipodal cell. Now this structure, central cell with this primary nucleus, and downside here, so two synergids are there. So this is one synergid and this is a fertilized egg. This is a spore, and one more degenerating synergy. So this one and this one, degenerating synergids. So antipodal cells also degenerate, then synergids also degenerate. This is a oospore or zygote, and this central cell, this cell is called as this cell is primary endosperm cell, and this nucleus, which is a product of triple fusion, that is called primary endosperm nucleus. Yes. So under this heading, first we are going to cover that is the endosperm. This endosperm develops from this central cell. Whatever central cell is present, where the primary endosperm nucleus is there, that endosperm develops from this structure. Now, so first one we are going to endosperm, second we are going to cover the zygote formation. So endosperm name is endosperm formation take place before the embryogen, or the endosperm formation precedes the zygote, means the embryogenesis. Zygote formation of the embryogenesis means, so Indian structure is the oospore, that is the zygote one. Before the zygote develop into embryo one, before that one the endosperm will form. Why means this endosperm is a nutritive tissue. So in the embryo ಬೆಳಿಲಿಕ್ಕೆ ಆಹಾರ ಬೇಕಲ್ಲ, so ಈ embryo ಬೆಳಿಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗಿದ್ದ ಮತ್ತೆ ಆಹಾರ ಪ್ರಿಪೇರ್ ಇರಬೇಕು. So food should be in the prepared condition. That's why endosperm formation take place before the embryogen. Yes. So now this endosperm is a nutritive tissue. Endosperm is a nutritive tissue for developing embryo. Hence, it is a product of triple fusion. It is triploid in nature. So this is a triploid in nature. So it is a nutritive tissue. So this is a nutritive tissue for the developing embryo. Whether the endosperm formation takes place almost all the family or genera, that is a major question. So this trip means double fertilization is unique phenomenon in the angiosperms one, but some family, exception theoretic, Orchidaceae, Podostaceae and Trapeaceae. Orchidaceae, Podostaceae and Trapeaceae endosperm formation is upset. Yes, so exceptionally ಒಂದು ಮೂರು ಫ್ಯಾಮಿಲಿ ಬಿಟ್ಟರೆ ಬಾಕಿ ಎಲ್ಲ ಎಂಡೋಸ್ಪರ್ಮ್ಸ್ ನಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಫಾರ್ಮೇಷನ್ ನಡೆಯಲ್ಲ. ಸೊ ಆರ್ಕಿಡೇಸಿ, ಪೋಡೋಸ್ಟೆನೇಸಿ and trapeaceae endosperm formation is absent. Now, based on the pattern of division, yes, so endosperm typification is there. So based on the type of division, so basically the endosperms are three types. Free nuclear endosperm, second one is a cellular endosperm, third one is a halobial endosperm. So that we are going to cover in this one. So typification means types types of endosperm. So this is about the brief introduction. But now we are going to cover the types here. Yes, students. Endosperms are the nuclear tissue. They are the product of triple fusion. They are the triploid in nature. The endosperm formation takes place before the embryo. Means embryo develop, but the pattern of division will decide the typification of endosperm. Endosperms are classified into three types. So in more division type names are. So this is the mature embryo set where this is the product of single, that is a zygote or oocyte, and this is the central cell. This is developed into primary endosperm cell, and this is the primary endosperm nucleus. Means this is the primary endosperm cell with primary endosperm nucleus. Yes. So this primary endosperm cell, ultimately this cell develop into endosperm. But the pattern of division is different. If the primary endosperm nucleus, in this case, first case, this is a primary endosperm cell with a single nucleus. This nucleus undergo repeated mitotic division. Yes, repeatedly mitotic division I do. But ಪ್ರತಿಯೊಂದು division ಆದಮೇಲೆ cell wall again means cell wall formation is totally upset. Means this division is a free nuclear division. Andre primary endosperm nucleus, first mitotic, second, third, and so on. So they undergo mitotic division and produce only nucleus in only single cell. Yes, initially this vacuole is central vacuole is present. Yes, so gradually the vacuole size decreases. Clear? So if the primary endosperm nucleus undergo repeated free nuclear means division, mitotic division without cell wall later on, then it forms the free nuclear, means this first type is called as a nuclear endosperm. Means in this case, repeated free nuclear division occurs. So this is called a nuclear endosperm. Yes, nuclear endosperm is seen in maize, rice, wheat, even sunflower, and one more particular name is given, that is the Cypsella bursa pastoris. So this name is mentioned in the most of the reference book. This is nothing but the shepherd pulse. Means E species of northeastern state ಅಲ್ಲಿ ಹೆಚ್ಚು ಕಾಣಿಸುತ್ತೆ. So nuclear endosperm means primary endosperm nucleus repeated mitotic division ಆಗುತ್ತೆ, but cell wall ಎಲ್ಲೂ ಆಗೋಲ್ಲ. So this is a nuclear endosperm. Yes. So later on, free nuclear division ಆಗುತ್ತೆ, ಪ್ರತಿಯೊಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಸುತ್ತಲೂ cell wall laid ಆಗುತ್ತೆ. So gradually it turns into different form. So initially it is a nuclear endosperm. So it is seen in the rice, wheat, maize, sunflower, even shepherd pulse, that is the Cypsella bursa pastoris. The next one, if the primary endosperm cell with the primary endosperm nucleus, the same case, this is the type second one. So after every cell division, after every nuclear division, if the cell wall is laid down, and then first mitotic division, cell wall is laid down, second mitotic division, again cell wall is laid down, and further every division, after every division, if the cell wall is laid down, so then every cell is surrounded by the cell wall. The second type is called the cellular endosperm. Means, can you differentiate this one? So nuclear endosperm ದಲ್ಲಿ ಒಂದೇ ಸೆಲ್ ಇದೆ, ಬಹಳ ನ್ಯೂಕ್ಲಿಯಸ್ ಇದೆ. ಸೆಲ್ಯುಲರ್ ಎಂಡೋಸ್ಪರ್ಮ್ ದಲ್ಲಿ ಪ್ರತಿಯೊಂದು ಸೆಲ್ ಅಲ್ಲಿ ಒಂದು ನ್ಯೂಕ್ಲಿಯಸ್ ಇದೆ. This is a multicellular form. So cellular endosperm is seen in petunia, then datura and endosperm. ಈ ಫ್ಯಾಮಿಲಿಯಲ್ಲಿ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಕಾಣಿಸುತ್ತೆ. ಸೊ ಬೇಸಿಕ್ ಡಿಫರೆನ್ಸ್ ಮೀನ್ಸ್ ನ್ಯೂಕ್ಲಿಯರ್ ಎಂಡೋಸ್ಪರ್ಮ್ ದಲ್ಲಿ ಸೆಲ್ ವಾಲ್ ಯಾವುದೂ ಡಿವಿಷನ್ ಅಂದ್ರೆ ಆಗುದಿಲ್ಲ. ಸೆಲ್ಯುಲರ್ ಎಂಡೋಸ್ಪರ್ಮ್ ಪ್ರತಿಯೊಂದು ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನ್ ಅಂದ್ರೆ ಸೆಲ್ ವಾಲ್ ಲೇಡ್ ಆಗುತ್ತೆ, ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. That's why the nuclear endosperm is a cellular endosperm. So third one is the hybrid, that is the feature between these two. It shows the intermediate character one. So the primary endosperm nucleus divides, yes, during the first division. After first division, cell wall is laid down. Illi cell wall I. Two cells are formed. The further division in both cases, in male cell and female cell ಎರಡರಲ್ಲೂ repeatedly free nuclear division takes place. Repeated division ಆಗುತ್ತೆ. So bari nucleus division, yes. So but the cell wall is laid down. After multinucleate condition is formed, then the immediately surrounding every nucleus cell wall is laid down. Yes. So initial first division is followed by the cell wall formation. Then many division is free nuclear division. After that, suddenly cell wall is laid down. This third type of endosperm is the third one. It is called as a halobial endosperm. Halobial endosperm. So nuclear endosperm, cellular endosperm, halobial endosperm. So this halobial endosperm seen in asphodilus, second one is the annulus. So ಎರಡು species ನಲ್ಲಿ halobial endosperm comes. So these are the types of endosperm. So but NCERT textbook only two explanations is given, but the heading cellular and nuclear endosperm ಕೊಡಲೇ ಇಲ್ಲ. So nuclear endosperm, cellular endosperm, halobial endosperm. So one more example is given in the textbook one, that is a coconut. When you observe the coconut, so this is a coconut. Yes. So this is a drupe type of fruit. So this is the epicarp, this is a mesocarp, and stony endocarp is there. Inside to this one, this is the epicarp, this is the mesocarp, this is the endocarp. This is nothing but the pericarp. This is the fruit wall. The endocarp is stony. Inside, initially, the tender coconut is filled with the water. Yes. Yes, water is filled. Yeah. So this water is nothing but the nuclear endosperm. The initial state, whatever the water present in the coconut one, that water is nothing but the nuclear endosperm. Yes. Later on, as the coconut matures, so this is the coconut, this is the endocarp. Yes. Inside, the white kernel will develop. Yes. Kobri in develop agatala. So we kobri noodle. Ilu ganji kata. The thickness of that kernel gradually increases. Initially, it is very soft one. Later on, the hardness increases. The tender coconut means the kernel will develop. So still it contains some water. There is some needle. Yes. So this kernel, again, kobri develop agila. that is nothing but the cellular endosperm. So in this base question, when you take the tender coconut where the water is present, water is nothing but the nuclear endosperm. The kernel, white kernel that will develop what the periphery. And then each cellular endosperm develop from the outside to inside. Origin the ಒಳಗಡೆ. So cellular endosperm develop. Coppery cellular endosperm. Neeru nuclear endosperm. So this is about the types of endosperm. Now whether this endosperm is completely consumed by developing embryo or not. ಈ ಪರ್ಪಸ್ ಏನಿತ್ತು ಎಂಡೋಸ್ಪರ್ಮ್ ಎದಕ್ಕೆ ತಯಾರಾಗಿತ್ತು ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ ಆಗೋತ್ತಿಗೆ ಅದಕ್ಕೆ ಆಹಾರ ಬೇಕು ಆ ಆಹಾರ ಕೊಡೋದು ಇದು ಎಂಡೋಸ್ಪರ್ಮ್ ದಟ್ಸ್ ವೈ ದ ಎಂಡೋಸ್ಪರ್ಮ್ ಫಾರ್ಮೇಷನ್ ಪ್ರಿಸೀಡ್ಸ್ ಎಂಬ್ರಿಯೋಜೆನೆಸಿಸ್ ಎಂಬ್ರಿಯೋ ಆಗೋಕ್ಕಿಂತ ಮೊದಲು ಈ ಪ್ರೈಮರಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಸೆಲ್ ಎಂಡೋಸ್ಪರ್ಮ್ ಆಗಲೇಬೇಕು ಸೊ ಈ ಎಂಡೋಸ್ಪರ್ಮ್ ಕೆಲವೊಂದರಲ್ಲಿ ಕಂಪ್ಲೀಟ್ಲಿ ಕನ್ಸ್ಯೂಮ್ ಆಗುತ್ತೆ ಕೆಲವೊಂದರಲ್ಲಿ ಕನ್ಸ್ಯೂಮ್ ಆಗೋದೇ ಮೀನ್ಸ್ ಇದೇನು ಎಂಡೋಸ್ಪರ್ಮ್ ಡೆವಲಪ್ ಆಗ್ತಾ ಈ ಎಂಡೋಸ್ಪರ್ಮ್ ಕನ್ಸ್ಯೂಮ್ ಮಾಡಿ ಈ zygote embryo development based on the usage means presence or absence of the endosperm in the seed one. Seeds are classified into two types. Yes, the types of seed we are going to cover now. So just I write downside here types of seeds. So here first one is endospermic seed अथवा albuminous seed. Yes, second one non endospermic seed or eggs albuminous seed. So this is an important one. Based on this question will arise. So examples type correct. So the endosperm is formed only for the to supply the nourishment for the developing embryo. Yes, the embryo zygote embryo develop ಆಗುತ್ತೆ endosperm complete ಆಗಿ ಯೂಸ್ ಮಾಡುತ್ತೋ in partially use ಮಾಡುತ್ತೆ. If you take the endosperm mixed with the albuminous seed, in this case, the endosperm is partially consumed. That's why small amount of endosperm still present in the mature seed. ಬೀಜ ಏನಾಗುತ್ತಲ್ಲ ಆ ಬೀಜದಲ್ಲಿ ಸ್ವಲ್ಪ ಎಂಡೋಸ್ಪರ್ಮ್ ಹಾಗೆ ಉಳಿಯುತ್ತೆ. Yes. So in this case, small amount of endosperm is present in mature seed. So in endosperm, small amount of endosperm is present in the mature seed. For example, this example theory. For example, coconut ತಗೊಳ್ಳಿ ಅಥವಾ maize castor. Yes, so even cereals also have the endosperm one. So ತೆಂಗಿನಕಾಯಿ ಇದೆಯಲ್ಲ ಆ ತೆಂಗಿನಕಾಯಿ ಗೊಬ್ಬರ ಇರುತ್ತೆ. So if you keep this coconut as it is, ಕೆಲವೊಂದು ತೆಂಗಿನಕಾಯಿ ನೀವು ಪೂಜೆ ಮಾಡಿ ಇಟ್ಟಿರ್ತಾರಲ್ಲ ಸೊ ಅಲ್ಲಿಂದ ಹಿಂಗೆ ಒಂದು ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ ಆಗಿ ಆ ಎಂಬ್ರಿಯೋ ಗಿಡ ಆಗುತ್ತೆ. Observe. So in coconut, in maize, in castor, small amount of or the endosperm is still present in the mature seed. Such seeds are called an endospermic seed or the albuminous seed. ಬೀಜದಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಇನ್ನ ಇರುತ್ತೆ. Eggs albuminous means in this case ಏನಾಗ್ತದೆ ಎಂಡೋಸ್ಪರ್ಮ್ ಪಾರ್ಷಿಯಲಿ ಕನ್ಸ್ಯೂಮ್ ಎಲ್ಲ ಕನ್ಸ್ಯೂಮ್ ಮಾಡಿ ಫಿನಿಶ್ ಮಾಡೇ ಇಲ್ಲ. ನೆಕ್ಸ್ಟ್ ಒನ್ ಇಸ್ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ or the eggs albuminous seed. So in this case endosperm is absent in mature seed. Why? So in mature seed ಅಲ್ಲಿ endosperm ಇಲ್ಲೇ ಇಲ್ಲ. ಕಾರಣ ಅಂತಂದ್ರೆ whatever the endosperm formed one, that endosperm is completely consumed by the developing embryo. Embryo ಆ ಎಂಡೋಸ್ಪರ್ಮ್ ಎಲ್ಲ ಕನ್ಸ್ಯೂಮ್ ಮಾಡಿ ಬೆಳವಣಿಗೆ ಆಗುತ್ತೆ. ದ್ಯಾಟ್ಸ್ ವೈ ಮೆಚೂರ್ ಸೀಡ್ ಅಲ್ಲಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಕಾಣೋದಿಲ್ಲ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ದಿಸ್ ಒನ್, ಈ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್ ಎಕ್ಸಾಂಪಲ್ ಇದರಲ್ಲಿ ವಟಾಣಿ ಇರ್ಬೋದು, ಪೀ, ಬೀನ್ಸ್ ಇರ್ಬೋದು, ಅಥವಾ ಗ್ರೌಂಡ್ ನಟ್ ಇರ್ಬೋದು. ಸೊ ದೀಸ್ ಆರ್ ದ ನಾನ್ ಎಂಡೋಸ್ಪರ್ಮಿಕ್ ಸೀಡ್. ಸೊ ನೀವು ವಟಾಣಿ ತೆಗೆದುಕೊಳ್ಳಿ ಅಥವಾ ಶೇಂಗಾ ತೆಗೆದುಕೊಳ್ಳಿ, ಅದರಲ್ಲಿ ಎರಡು ಕೊಟಲಿಡ್ ಅಂತ ಕಾಣಿಸ್ತದೆ, ಎಂಡೋಸ್ಪರ್ಮ್ ಇರೋದಿಲ್ಲ. ಸೊ ದೀಸ್ ಆರ್ ಸಮ್ ಎಕ್ಸಾಂಪಲ್ಸ್. So types of endosperm means three types are there: nuclear endosperm, cellular endosperm, alluvial endosperm. Based on presence or absence of the endosperm in the mature seeds are classified into endospermic or the albuminous seed, where the small amount of or the endosperm is still present because it is partially consumed by the developing embryo. Example: coconut, maize, castor. So if the endosperm is completely consumed during the development of embryo, mature seed ಅಲ್ಲಿ endosperm ಕಾಣೋದಿಲ್ಲ. Such example: pea, beans, even the groundnut one. So in that one only cotyledons are visible, not the endosperm. So this is about the endosperm. ಈಗ again primary endosperm cell along with primary endosperm nucleus divide ಆಗಿ so ಇಲ್ಲಿ. So this is the primary endosperm nucleus. So this will develop into endosperm. Yes. So in endosperm this endosperm is consumed by the developing embryo. A zygote in the embryo will be the endosperm consumer. So this is about the first structure comes under the post-fertilization structure and event. That is nothing but endosperm. Yes. Now the next topic is the embryology, means types of embryo. Means basically it is divided into monocot embryo and dicot embryo. In that initial development of embryo pattern is almost similar. Later on embryo development pattern differ in the monocot and dicot. The embryogeny of the second one, embryo structure, we are going to cover after it. Yes students, now the next topic is the formation of embryo. So just 5 minutes back we have seen how the endosperm development takes place, type of endosperm and the type of seeds based on the presence or absence of the endosperm, whether it is albuminous seed or the ex-albuminous seed. Now next one is one more structure, that is a post-fertilization structure, that is nothing but development of embryo. Yes, so you take the double fertilization. So this is the central cell with the primary endosperm. This is the primary endosperm cell with the primary endosperm nucleus that develop into endosperm. Just we have seen the endosperm formation take place before the zygote embryo formation. Now comes to the function of zygote. This is the product of syngeny, which is a diploid in nature. This is a primary endosperm cell is a triploid. The endosperm is also triploid. Zygote is a diploid. This is also called as oospore. The development of embryo from this zygote is called as a embryogeny. So this is called as a embryogeny. So embryogeny is how the embryo development takes place. But the pattern of development of embryo in monocot and dicot is totally different. But initially it is same one. Yes. So this is a zygote. Now see how changes will occur. So some figures are there, some more figures I am going to draw. So this is a zygote. Yes. So which is a diploid in nature, which contain both maternal and paternal carrier. Now this zygote undergo first division and produce two cells. So now this is a micropylaryn. Yes, this is a chalazarin. Clear? So this side is the micropylaryn, this is chalazarin. The cell toward the micropylaryn is called as a suspensor cell. This is suspensor cell. The cell present toward the chalazarin, this is called the embryo cell. This blue color cell is an embryonic cell. This is suspensor cell and embryo cell. In both the dicot and monocot, the zygote divide and produce the two cells. suspension cell and embryo cell. Now first we will study the dipole pattern. Here the suspension cell undergo repeated division. So this black color cell undergo repeated division and produce the suspension. Yes, so this complete structure is called the suspension. The large cell, this is called the hostoria or hostorium, and this the first cell is called the hypophysis. Hypophysis. This hypophysis, this first cell which is near to the embryonic cell, this hypophysis later on develop into radicle. This cell, this hypophysis develop into radicle. So this complete structure, the black color structure, is called as a suspension. So suspensor. Here. Now this embryonic cell, this cell undergo two vertical division. Okay? First division is this one, and one more division is a 90 degree to this one. So and one transverse division occurs. So totally 8 cells are formed. 4 cells above, 4 cells below. Yes. So this lower cell, this cell are called the epibasal cell. Epibasal, and these cells are called the hypobasal. Yes, epibasal means so main end, hypobasal means kelding. So ultimately the globular structure is formed where the 8 cells are present. This structure almost globular embryo theory. Yes. Now this complete structure undergo repeated division in end cell. So this epibasal hypobasal means this epibasal cell in future. So in epibasal cell in this cell will develop into plumule. Yes, and cotyledon. ಈ epibasal cell ಮುಂದೆ ಹೋಗಿ plumule ಮತ್ತು cotyledon develop ಆಗುತ್ತೆ. Hypobasal develop into hypocotyledon. Yes, so this will develop into hypocotyledon. Understood? The zygote divided into 2 cells, one suspensor cell, one embryonic cell. Suspensor cell divide repeatedly and form the suspensor. The last cell is called the hostorial cell, and the first cell is called the cell near to the embryonic cell is called the hypophysis. This hypophysis cell in future develop into radicle. This embryonic cell divide into 2 vertical divisions, one like this, and one transverse division. Totally 8 cells are formed. Now this 8 cells, lower cells are called epibasal and these cells are called hypobasal. Here, epibasal cell in future develop into plumule and cotyledon. Hypobasal develop into hypocotyledon. Now in this embryo, repeated division takes place and different layers are formed. So when you observe, so this outer cell is something different and this cell is different. So this is called the globular embryo. Now this embryo is called a globular embryo. Here some differentiation occurs. So the outermost cell, this is called the dermatogen. In organ cell, this is the dermatogen that will produce the outer layer, epidermis one. So inside this is the ground tissue. In that one, further development means differentiation takes place. Again, meristematic tissues are produced. So this is a globular embryo. Gradually, this globular embryo turn into a heart-shaped embryo. So this is heart-shaped embryo. Yes. So now this outer cells are called the dermatogen. This is called the prolemma, and this is called the ground meristem. Yes, gradually globular embryo turn into heart-shaped embryo. Still suspensor is present, which is embedded in the micropylar region. Yes, so micropylar region means this is the micropylar region. Illintra suspensor, the nutrient. So this is the still heart-shaped embryo. Now this heart-shaped embryo develop into mature embryo, means completely developed embryo will appear like this. So this is a suspensor, I will draw a little bit shorter, and this is a limuli structure, and cotyledon I will show with a different color. So this is a mature embryo, fully formed. Yes. Now if you observe this one, so this is as usual suspensor and suspensor cell. This is a root cap. This is root, and this complete structure is called the radical filament. So this is the radical. This radical developed from the hypophysis one, and this blue color structure which is present in between these two. This is called as a plumule or the shoot apex, and this two color structure, this one and this one. Yes. So these are cotyledons. These are cotyledons. Yes, two cotyledons are there. So this is the plumule. Now the region of the embryo from the attachment of cotyledon, E portion in this region above the pattern of this one. So this region, for example, if you take the same embryo, so same embryo diploid embryo. So these are the cotyledons. Yes. So this is the plumule, and this is the attachment of plumule with the cotyledon, and this is the radicle. Yes, ಅದೇ embryo ನೀವು open ಮಾಡಿದ್ರೆ elaborate retouch structure. The region above the pattern of alignment, cotyledon will attach it. The above region is called the epicotyle, and region below the attachment of cotyledon is called the hypocotyle. So this region means this region is called as the epicotyle, and this region is called as the hypocotyle. Yes. So ಇಲ್ಲಿ the plumule is there, just beside this one cotyledons are attached. The region below the point of attachment of cotyledon is called as hypocotyle. The region above the point of attachment of cotyledon is called the hypocotyle. Yes. So this is a radicle or the root tip covered by the root cap, and these are the two cotyledon, and the plumule is a shoot apex. So this is a mature embryo. That embryo developed from this zygote. Now when you take a monopod one, the initial development is same here. So this will also, the zygote divided into two cells, suspensor cell and embryonic cell. So this is a suspensor cell. Yes, this is the embryonic cell. This embryonic cell divide again and produce two cells. This is called the middle cell, and this is called as the terminal cell. So ಈ ಕಡೆ ಇದ್ದು terminal cell, ಮಧ್ಯದೊಳಗೆ ಇದ್ದು middle cell. Now the terminal cell, this cell in future develop into again plumule and cotyledon. ಈ terminal cell ಇಂದ plumule ಮತ್ತು cotyledon ಬರುತ್ತೆ. Middle cell ಇಂದ almost radical ಮತ್ತು hypocotyle develop. Hypocotyle means ಕೆಳಗಿನ portion. Now this cell ಏನಿದೆಲ್ಲ, this cell undergo repeated division, again it produce the globular embryo. Globular embryo, and this is the suspension. Yes, this is suspension. So here also again differentiation takes place. Outer layer is the protoderm, means this one dermatoderm. Inside ground tissue is there, and some meristematic tissue is present. So like this one. So gradually, in this case, initially two cotyledons are there. Out of this one cotyledon express in exaggerated manner, and that will push the plumule towards the one side. Embryonic axis one kade push ಮಾಡುತ್ತೆ and ಮತ್ತೊಂದು cotyledon elaborate ಆಗೋದೇ ಇಲ್ಲ. So ultimately the mature embryo of monocot will appear like this. This is little bit complex structure. Just try to understand. So this is yes. These structures are difficult. Let's try to draw this figure 1 or 2 times, and then it will be familiar for you. So you take a monocot embryo. Almost parts are same only, but some 2-3 parts are some extra parts are there. So try to understand this. Now this is a complete embryo. So this is in this case, two cotyledons are there, but only one cotyledon is express one more cotyledon is a rudimental one. So this structure is the actual cotyledon, that is called the scutellum. This structure is the scutellum one. This is actually one cotyledon. One more cotyledon is a smaller one, that is called an epiblast. You have seen, this is the one cotyledon which is a larger one, which is a shield-like structure. Another cotyledon is a very small, which is called an epiblast, which is just present for the one side. So due to development of this cotyledon, the embryonal axis is pushed towards the one side. This is the embryonal axis. Yes. So actual cotyledon is called as scutellum, and this is the plumule. This is the shoot apex or plumule, which is covered by the almost the special tissue that is called polyptile. Yes. So yes. Now this is called as a radicle. This is a radicle, and it is also covered by the special tissue that is a coleorhiza. Yes. Now this is a point of attachment of cotyledon one. The region of the embryo from the point of attachment of cotyledon, the region above, this is called as the hypocotyle, the epicotyle, and this is called as the hypocotyle. This is epicotyle and this is hypocotyle. Yes, when you take the type of embryo, so this is the embryonal axis, this is the plumule, this is the radicle. These are the two cotyledon. This is the point of attachment. The region of the embryo above the point of attachment is called the epicotyle. Same here also epicotyle is there. So region below the point of attachment is called the hypocotyle. Difference is here shoot apex or the plumule is covered with a special tissue that is called the coleoptile. This is one tissue, and the radicle or the root tip or the root cap is covered by the special tissue that is called the coleorhiza. So these two tissues are seen only in the monocot embryo, and the actual cotyledon is called as scutellum. This is the actual cotyledon. So based on this one, question will come. So cotyledon in the monocots is called as scutellum. One more cotyledon which is not elaborated, that is called an epiblast, which is present towards the one lateral side. So this is about the monocot embryo, and this is about the dicot embryo. So the embryo developed from this zygote. For this development, nourishment is required. That's why the endosperm formation takes place before the embryo development. This pattern of development of embryo is called as an embryo gel. Yes, so this is called the embryogeny. So these are the two structure comes under the post-fertilization structure and events. One embryogenesis, embryo ಆಯಿತು, ಎರಡನೇದು endosperm formation. Now come to the last part of this chapter. Only seed, fruit, then one more topic is there, that is about the apomixis and the polyembryony. All this topic we are covering in the next video. ಅಲ್ಲಿವರೆಗೆ ಎಫರ್ಟ್ ಮಾಡ್ತಾ ಇರಿ, ಸ್ಮಾರ್ಟ್ ವರ್ಕ್ ಮಾಡ್ತಾ ಇರಿ. ಟಿಲ್ ದೆನ್ ಟೇಕ್ ಕೇರ್, ಗುಡ್ ಬೈ, ಹ್ಯಾವ್ ಅ ನೈಸ್ ಡೇ.